Hello, everyone. Welcome to Nano Talks. Sorry for a few minutes of delay. I am Fabio Pulizzi. I'm Chief Editor of Nature Nanotechnology. With me, or virtually with me, is Melissa Plail from Nature Communications, and we are co-hosting today's webinar. And we're talking about nanoplastic. This is a plastic in general is a topic that often appears even not just on scientific papers, but more and more in the news in general, and plastic pollution and the potential problems that it can cause and how to get rid of it, what to do about it. So we have 4 panelists today. I should say that Nature Nanotechnology has published recently 2 perspectives, and inspired by these 2 articles, we have decided to have a discussion about the topic. And our 4 panelists are Denise Mitrano from ETH. Denise has worked for many years on nanomaterials and their environmental impact. Similarly, I would say Julien Gigault, who is now based in Canada, Université Laval. Julien, correct me if I'm wrong, but he's also associated with CNRS in France. And then we have Nanna Hartmann from the University of Technology of Denmark, and Martin Wagner from the NTNO in Trondheim, and both experts of nanoplastic, plastic in general, and nanomaterials. We want to discuss with them several points. There's first of all, there's something that we're wondering several times, and what is nanoplastic actually? Does it make sense at all to distinguish nanoplastic from plastic, from microplastic? Is there actually a geometric difference simply, or does it make a difference for the environment and even for policy? And other points of discussions are, how much do we know actually of how much plastic is, how dangerous plastic is for the environment and for humans even? And how do we know how to detect it? Do we know how much there is out there? And do we know how to see it? How to actually monitor it in the environment? Melissa, if you have anything to add, please go ahead. Otherwise, we can start.
Yeah, I think you've covered it all. Obviously, it's a really important topic that many of us are interested in finding out how much nanoplastic there is in the environment, and just really what the difference is between it and microplastics. So I think we can start.
Okay, so Melissa, you have the control, and so these are some of the points that we want to discuss, and I would just leave it for any of the panelists to start discussing what they can tell us about nanoplastic and the definition of it.
Should we just go to the next slide, and I can jump in with kickstarting the discussion? Or
thanks, Nanna.
Yeah, So thank you for allowing me to open this discussion on defining nanoplastics, even if I have not been an author on one of the 2 recent perspectives, but I really enjoyed reading those. Defining nanoplastics, you can say that for nanomaterials, we've had this discussion going on for more than a decade now, and I think this discussion is still ongoing. So I think we can have a long discussion also on how to define nanoplastics. I enjoyed reading both the perspectives by Denise and Julien and the co-authors there, which I thought gave some really interesting food for thought and reflection. So I'll just take the liberty of highlighting a few aspects from your perspectives. So in Denise's paper, you argue that size categorization of plastics into micro and nano, you say that it might be counterproductive and more a question of convenience. And that size is a continuum, so why should we set any cutoff values? And Julien, in your paper, you argue that definitions should be characteristics based, and I know that you will come back to that in a slide or 2. So I just want to start off by saying that I agree with both of you and your viewpoints to a very large extent as a scientist. But I also have a more regulatory training or background, which may color my view in terms of the need for pragmatism. So what you see on this slide here is from a paper that I published also together with Martin back in 2019. And I think it kind of reflects also in that paper that like my belief that as a scientist, we cannot live in a vacuum, but we need to provide inputs also into regulation to also increase the science-based foundation for environmental regulation. And sometimes, or maybe often, cutoff values, they are needed, especially for regulatory purposes. And I just want to give a little simile. So you can say that there is a regulation saying that you have to be 18 years old to vote, right? So that's when you're categorized as being an adult. And even if we can say that age is a continuum, so why exactly should we say 18 years of age? Or maybe it would be better to look at maturity as a characteristic. So in that sense, I can completely agree that theoretically, the most correct way of characterizing might be based on those factors, or based on maturity, maybe, but it's just not very practically feasible. So in our paper from 2019, we argue that size-based categorization is needed for regulating nanoplastics and microplastics, and also for monitoring purposes. But we also acknowledge that the size cutoffs, they will be arbitrary, and they are not biologically informed, because size is not the only parameter that is affecting biologic interactions. So that's why we chose to simply adhere to the SI units, and that's what you see in the slide here. So we say nanoplastics is between 1 and 1,000 nanometers, and microplastics, I believe, between 1 and 1,000 micrometers. I would actually argue, on the other hand, that if we talk about a 5 millimeter cutoff limit, that kind of gives some kind of implication or perception of a biologically informed cutoff value. That's why in this paper we decided that to go for the very pragmatic solution and say, okay, we stick to the pragmatism, and we say we adhere to the SI units. So I would like to maybe also pose a question that is like, do definitions and size cutoffs, do they really impair our work as scientists? Why not just accept that there is a categorization framework that is needed for regulatory purposes, and then we as scientists can do whatever we want in the laboratory? So that's kind of a question that maybe we can discuss. And then if we go to the next slide. As another opening remark to this discussion, I would just like to say that size is not the only property that can or should be considered in the categorization framework. There are also other characteristics that we need to look at. For example, shape that also exists in a way as a continuum. You can say there is a continuum from a sphere to a fiber. And maybe more importantly, it's important to it's less meaningful or less important to discuss what is nano, but more important to discuss what is actually plastic. And there we also have a continuum from a natural polymer to a synthetic polymer. So I think those were just my opening slides.
Thank you.
I will take the next slide. So thank you, Nanna, for this very, very great introduction. So of course, I think size is always needed, to do some like regulation policies, and we need parameters to define how much we have in this category. But I will not debate about the size definition, because I think it's infinite debate. So when we decide to write this perspective, we realize different things. The first thing is about the definition and the analytical techniques to characterize a nanoplastic, and we realize that when we give a cutoff of this particle in terms of size or shape, some bias can occur in terms of what kind of techniques I will use to characterize my submicron nanoplastic or microplastic. So we decided to define nanoplastic as a series of properties rather than just to give a size or a size limit, a size cutoff that can be very arbitrary. But I think it is necessary for at some point for regulation, like protection for the environment, for health, and other issues. Also, what we realized that there is a shift in terms of terminology. You see where I'm going, but the last 30 years, a lot of work have been done about nanometrological development for characterizing nanoparticles in the environment. I think Nanna, Martin, Denise, you all participate to different techniques development to characterize nanoscale materials in the environment, and realize that polystyrene nanoparticles at some point they were largely used in this field to calibrate method, to calibrate like experimental setup and designing an experiment for investigating the fate or toxic effect about nanoparticles. And when we plot this number of publications just by changing the terminology about particle and polystyrene and nanoplastic or macroplastic with polystyrene, we realize that we clearly see this this shift of terminology. That's not bad in that sense, but some brand new method can be used as standard characterization or standard method, but this method was were already like developed or investigated in the past for different kind of particles. So by writing this perspective, we also want to say that some methods are very useful and are used for nanoparticle engineering nanomaterials that can be translated to nanoplastic, and it's for what at some point we would like also to address this this common point between nanoplastic and macroplastic, or the diversion point, sorry, and also with engineering nanomaterials that we think can be also can induce some mistake about the definition of nanoplastic. But all these properties definition that we summarize in 6 different kind, which mostly is a Brownian motion for us, is one of the main definition for colloidal particles. Also, the ability to interact with organic matter, the natural organic matter, or other colloids in the environment, because considering nanoplastic as only one particle in the environment, I think it's very it's reducing the problem, and it's not very accurate in terms of environmental relevancy. So we have to consider the transformation pathways in the environment, including this colloid, other colloid system. So we try to put these 2 properties in this as a main definition, and then to discuss about the interaction with the biota, how nanoparticles or a nanoplastic can be different in terms of assimilation with the biota. But this is Denise did very good work also on this aspect, so I think she will talk more about that. And also in terms of detection, because this is a point, right? We want to detect and analyze nanoplastic in the environment. So for now, we still start. We have some idea, which technique we will use, but it's very important that you cannot visualize nanoplastic compared to macroplastic. This is very important to distinguish that the size regarding the wavelength of the incident light is governing the ability to detect the particles. So for nanoplastic, we clearly are below this wavelength. So we have to develop new techniques, new methods to catch up or to characterize and quantify, because I think this is a second challenging analysis and identify and quantify nanoplastic environment. So this is the sixth bullet point that we would like to highlight in this perspective, and if what I decided to put this box in this slide, and I will be happy to discuss about that. review.
Yeah, thanks, Julien, and also Nanna for your perspectives. I mean, I think that, well, I don't know if we want to go back to the first questions or stick on the slide, doesn't matter, but it's still about the definitions of nanoplastic, because like Julien and his perspective, we didn't really want to jump into the fray of trying to suggest what our own definition was, and we kind of got around this in a section of our perspective called a nanoplastic by any other name is still a nanoplastic. Maybe it's my Shakespearean poet in me that just says it like this. But I still do think that, as Julien and Nanna both said, that it's really about the way that the particle behaves in the environment, which is really what we should be focused on, and not necessarily trying to define a limit or a definition for definition's sake. Although I can totally agree that in terms of policy and regulation, definitions are very, very important. But if we also look back to engineered nanomaterials, where all of us have previously worked also in our former life before plastic, we can see that definitions aren't always fixed there, even in terms of policy. So Antonia Praetorius also published a perspective a few years ago in Nature Nanotechnology, which also looked at the regulatory definitions of engineered nanomaterials in different sectors. So for like cosmetics or agrochemicals or different types of things, and even regulations from different sectors all within the EU had a different definition of nanomaterials, and that also had policy consequences. And importantly, in her perspective there as well, which I think equally applies to what Julien was saying about how can actually measure these nanomaterials, some of the definitions which were prescribed were not actually physically measurable from an analytical chemistry point of view. And so if you make a regulation or a definition of a material, but you don't have a way to actually measure it, it's not a very useful regulation, in my opinion, because it's not really measurable or enforceable if you can't actually measure it.
And just jump in here. I mean, I think pretty much we basically agree that like defining stuff does matter, yeah. And I would just add, it doesn't only matter for regulatory purposes. I think we're really fixated on regulation sometimes. I think it also matters for us as academics, right? Terminology really matters, and I think Julien did a good job in showing that just if you change the search terms in your literature search, you get all of a sudden you capture older literature on nanoplastics that we hardly read simply because we're searching for nanoplastics and not for nanoscale polystyrene particles. So I think it really matters in the way that we as academics kind of dig into the available knowledge. So I think that's quite important for us as well. Terminology changes over time, and I think that's something that is also true for regulatory definitions, right? It should be flexible, and that's something that we highlight in our paper, that with evolving knowledge, of course, with better detection methodologies, etc., of course, those definitions could be really flexible and could be adopted. Being a biologist, I think about size in a very different way, not about like the analytical techniques to measure it, but I'm thinking about what is the biological rationale for size cutoffs really. And the only thing that really comes to mind, if you think about it from a toxicological, biological perspective, is translocation. So there's this idea that at a certain point, at a certain size limit, particles can cross biological barriers, and they will end up inside an animal or an organism, and they will end up maybe inside of tissues and maybe even inside of cells. So I think like we could aim for developing a biologically informed size cutoff there. And we really don't have a lot of knowledge on that, but I would say then the size cutoff would be at 10 micrometer, because we know from a couple of studies that particles below 10 micrometer have an increased tendency to translocate to tissues, at least in mammalian species. We don't really know about all the invertebrates out there, so there's a big knowledge gap in there. But if we would really go for a biological informed size class, then my bet would rather be 10 micrometer downwards. Yeah, so that would a bit change like the whole discussion, because then you might not call it nanoplastics anymore. But that's just to shake up the discussion a bit.
I think that brings us nicely on to the next part of the discussion, which is what are the potential dangers of nanoplastics? So who would like to go first with that?
Well, I think that this also relates a lot to what Martin was just saying, and then this feeds, I think, quite nicely into this slide that we had presented also in the recent perspective, looking at really what functional changes are happening when particles of different size classes are either interacting with a biological organism, like we can see in the top half of the slide in the green bars, or in the bottom half of the slide in terms of chemistry and transport of particles in different environmental systems. So when we think of biological interactions, I mean, this can either be a response or a biodistribution within the organism. And here in the gray bars that go vertically throughout the figure, we relented to show the two different aspects of nanoplastic that Nanna had also presented in the first slide, of either 1 to 100 nanometers or 1 to 1,000 nanometers. And we can see that particles of different sizes will have really different possibilities in terms of where they're able to go, either in a biological system or be transported through the environment. And some of these aspects may be specific to plastic, and some aspects may not be relevant to plastic. So in some of the smaller gray bars, for example, this oxidative stress or solubilization, these we put in there because we found them to be important when we looked at engineered nanomaterials, especially inorganic engineered nanomaterials. These can dissolve or cause oxidative stress, but these probably won't be as relevant or relevant at all for plastics, depending on the plastic type. So in this case, we will be able to look at a certain size of material and already be able to estimate what might be the biological interaction or the transport, and which factors might not be able to be well transferred from our knowledge from engineered nanomaterials as we work in this new field of plastic.
Yeah, I added this slide, but it was mainly to highlight some past efforts that had been to try to look into, is there actually a specific effect or a larger effect of the smaller particles. So this is from the Gesamt report in 2016, where they saw some indications that the smaller particles had more severe biological effects. But it's yeah, it was just actually leading up to the next slide for Martin.
Yeah, I think it's like a common assumption that we often hear also in the scientific community, that the smaller particles get, the more toxic they are. I think that's like a default assumption that we are having, and again, that's, I believe, based on the idea that like if you deal with smaller particles, then you get a higher potential for tissue translocation, basically, I believe. As you all know, the research on nano and microplastics has exploded over the last couple of years, really. I think like all of us would agree that we can't really keep up with the papers that are being published on a daily basis. It's very hard really to know what's going on, because you see at least like 20 papers published per day nowadays. So it's really, and I think it's fantastic that we are seeing this expansion in research and this focus on nano microplastics and plastic pollution in general. And that was kind of an idea that we followed up in recent work that we did for the Californian government, where the Californian colleagues, they compiled a database on toxicity data for nano and microplastics, and they were amazing. They mined like data from 167 publications, looked for toxicity data of nanoplastics and microplastics from 80 species. We have more than 6,000 data points on the toxic effects of those plastic particles in there. And that's like all in a Shiny app, and hopefully soon to be publicly available for everyone to use. I think it's a great tool really to look into what kind of toxicity data do we have on plastic particles. And I just took the liberty to look and compare the toxicity of different sizes of plastic particles. If you can move to the next slide, because I wanted to really address or investigate whether, like, if we do a meta-analysis of the published data, whether we can really support the idea that nanoplastics are more toxic than microplastics. And it really depends on how you look at the data. I'm trying to talk to you rather quickly through this. So you're seeing species sensitivity distributions plotted over here in the colorful dots there. And that's basically data from the literature, and it's showing you one organism, and it's showing you the lowest concentration that has of a plastic particle that has a negative effect in that species. So all of the little colorful dots is one like species, aquatic organism from a toxicity study, and then that's the lowest concentration where that particle had a negative effect on that organism. So basically, if you look at the lower left of all those size distributions, those are the most sensitive animals, the most sensitive species to the most toxic plastic particles, so to say. So what I did here, I split it up in different size classes. So the blue dots that you're seeing in the graphs are microplastics, larger than 1 micrometer, anything. The yellow dots that you see here is what we would consider submicron plastic particles or nanoplastic particles from 100 nanometer to 1000 nanometer. And the red ones are the actual nanoplastics from 1 to 100 nanometer over there. So if you look at the left graph over there, I've plotted the concentration of plastic particles per milliliter of water. And what you're actually seeing is that microplastics, the blue dots, are much more toxic than nanoplastics, the red dots, when you plot it as this kind of numerical concentration. And that's kind of counterintuitive because we believe that nanoplastics would be more toxic. But if you compare microplastics and nanoplastics on this kind of particle count per volume basis, you would actually say that microplastics are more toxic to aquatic organisms. And that kind of makes sense because most of the toxicity studies that we're seeing, they use mass-based concentrations, which I'm showing you in the middle. And then, of course, if you use the same mass of a particle and you have nanoparticles, you have just much more nanoparticles than microplastics in there. So if you plot the same data on a mass-based concentration graph in the middle, you see that this kind of difference in size and toxicity kind of disappears. So it looks like that, like the middle fraction, the 100 to 1000 nanometer particles, they are quite similarly toxic to the nanoplastics, 1 to 100 nanometer. And the microplastics, the blue distribution that you're seeing here is also kind of aligned with that. So one could argue that the better metric to compare the toxicity of different size particles would maybe be the mass per volume. But maybe not. If you think about it biologically, maybe neither the particle count nor the mass of a particle, of a plastic particle matters. Maybe it's really the volume. If you think about that animals ingest plastic particles and that those particles occupy space in the gut of those organisms, then it might be rather the volumetric concentration that matters here. And that's what I'm showing you in the graph on the right side of the presentation over there. So if you plot it as volume per volume water, you see actually that pretty much the microplastics, blue distribution, and the nanoplastics, the red dots, they align pretty much. So it would really be the volume of the particle that matters. However, you see that in between the yellow particles, 100 to 1000 nanometers, on that kind of dose metric, they seem to be more toxic than the other two dose categories. I'm showing you this not only to confuse you a bit, but mainly to tell you that it matters on which metric you compare the negative effects of plastic particles. Is it the count? Is it the number of particles? Is it their mass? Is it their volume? Is it their surface area? We had that discussion for engineered nanomaterials, showing that if you inhale particles, it's really the surface area that matters. So I think we really need to discuss what's like the best way of comparing different size classes with regards to their negative effects. And that's something that we as a scientific community need to do. Another thing that I want to highlight before I quit here is if you look at the left graph again, that's the particle number per volume. Right? So that's the concentration over there. And if you do this species sensitivity distributions, you see that you end up at quite low levels of plastic particles in the environment that would have a negative effect on very sensitive aquatic species. And this kind of tells you that as we are compiling more and more toxicity data, we are getting closer to defining what threshold it is really that certain particles have for evoking negative effects on biota in the environment. Stop with that, just some food for thought here.
Thanks, Martin. As you said, this could add. It certainly shows us that it depends how you define things. Sorry, Nanna, you've got your hands up.
Yeah. No, I'll. Okay, I'll go ahead. Yeah, no, thank you for sharing these data, Martin. I agree that well, definitely it gives food for thought and maybe also a little bit of confusion, or at least yeah, I've been thinking a little bit about these graphs now. And I was just thinking, because if you look at these graphs, then you have every point that is representing a dose response curve. So basically, for every single dot, behind every single dot, there is a dose response curve with an x-axis, meaning a quantification of exposure. And I think that one thing that we have learned from nanomaterial research is how hard it can be to control and to measure and to describe exposure in a meaningful way. So exposure, should it be expressed based on primary particles or agglomerated particles? So I think maybe instead of asking how far are we in understanding nanoplastic effects compared to microplastics, I would also pose the question, how far are we in performing meaningful and reliable tests? Because I think that's something that comes from the environmental nanomaterial research, is that, you know, we have been challenged with the existing test systems that were originally developed for soluble particles, and how do we soluble chemicals, and how do we adapt those to now be testing particulate material? And also, have we successfully transferred the lessons that we have learned from nanomaterial research into this new plastic field? And I know that, Julien, in your paper, that you're also recommending for, I think you're saying that you should have visiting nanomaterial scientists into the plastic labs to ensure this transfer of knowledge from this field. And I could also maybe just share that I had a collaboration with Anita Yemek last year, where we wrote a paper about the quality of studies on like polystyrene ecotox studies, like what is the quality of the reliability and quality of data there. So we use different sets of criteria, so the Guide Nano and the Dana criteria, and we gave scores to how well these studies performed in terms of different checkpoints. And actually, we saw quite low scores, especially for substance characterization. So I think maybe we still have something to learn in terms of like that we utilize all the knowledge and all the experience that we have gained from nanomaterial research in making sure that behind each point on these SSDs, that behind each point there is a meaningful data set with a meaningful x-axis.
I think I would just jump in here, Nanna. I think it's very salient points you're making. We started this whole research, we as a community, I think, with very crappy studies, to be honest. And I'm kind of a bit apologetic about it, because I felt like I was new to that. I have never done engineered nanomaterials before in my career, so I was really fresh to that. And then when you get a new stressor, you get a new pollutant, you just apply the stuff that you have at hand from chemicals, and we slowly learned that that's maybe not appropriate. So we can't just use like the standard setups and for the toxicity studies that we have for dissolved chemicals. Just put some nanoplastics in and then everything's great. I think that's what we're slowly learning as a community. I think we've made quite some progress in the quality of toxicity studies. That's my personal opinion. Would be nice to see if that like translate to better scores when you do quality assessments over time. I haven't seen any analysis on that, but just my personal impression is that we are improving on the experimental design. With regards to what can be learned from engineered nanomaterial world, I would just make a comment saying that yeah, some of us have been worked on that before, so that really facilitates like the transfer of experiences and maybe improves experimental designs. But I also want to highlight maybe just like precarious conditions, working conditions for academics, right? So in my feeling, many of the people that did nanotoxicology that I know from before just have left academia, and a lot of experience is gone with them. So that's just to throw in maybe another dimension here.
But I would wonder if people are leaving academia for which purposes as well. I don't think that they leave because they studied nano, you know?
Maybe. No, but
that might
no,
be
but
unrelated, I
yeah,
guess, in
but I
all
think like,
fields, people leave.
but we've been seeing, you know, like, and that's what we talk about plastics as well. We've been seeing for nano, we've been seeing the spike in funding and spike in publications, and then that peak like really went down again. So I think like a lot of the funding for engineered nanomaterial science like just disappeared, and that forced people to either move on to another topic, that being plastic pollution, or just getting a job outside of academia. So I think like we are not doing our best in a sense that we are not transferring knowledge easily from that topic to this topic.
I would say yes and no, because I think a lot of us have, I mean, me, myself, Julien as well, have transferred knowledge. And even when you look at engineered nanomaterials, like my PhD advisor was looking at natural colloids before. So it was really looking at particles throughout, whatever the chemistry of those particles were in the context of those particles. It was really colloidal chemistry and how particles behave in the environment or in or interact with organisms. But I think when you talk about like a boom of funding for engineered nanomaterials and now a boom of funding and papers for plastics, maybe if you also looked at how many PhD students there are generally compared to postdocs, compared to assistant professors, I mean, I think that the field of academics generally is unfortunately this pyramid anyways, right? So maybe it's many people would have naturally moved on to a different topic or a different position, regardless of whichever topic they were studying during their PhD or postdoc.
Right, I think it's more general problem.
I hope it wasn't like a black hole for people that were studying engineered nanomaterials, the black hole of plastic now either. I think that there can always be transferable skills, even if you work outside of academics and no longer work in plastic, because this sort of knowledge can be used in a lot of different contexts.
Right.
I had a very specific question about this, still in the comparison with engineered materials. So Martin, you showed us this data, and I find it extremely interesting actually to show how comparing different sizes with different types of concentration. That gives you a different topic. Do you know if for engineered nanomaterials we have similar things? We have similar comparisons? I mean, I'm aware that also in the field of engineering, also when looking at toxicity of engineered materials, there has been for sure debate on what do you actually look at. Where do you look at mass or volume, or perhaps maybe there was some kind of consensus forming. But do we know if there is some type of study like this as well?
Well, I think like for inhalation toxicity, I'm not an expert on that really, but I think like for inhalation, there's kind of a consensus in that air pollution community that really the surface area matters. So they don't really look at particle size. They don't really look at those types of parameters. They really look at the surface area. And I think it should all go back again to be biologically informed. So it really depends on what type of mechanism of action you would expect. So if you think plastic particles are just like diluting the food that an animal is ingesting, then it would be the volume that you would be interested in. If you think like their surface is interacting with a biological entity, with the tissue in the lung, with an epithelium, then it should be like the surface area, right? And then the charge becomes important and things like that. So I think we should aim for a biologically informed dose metric or a relevant metric here. And the problem is that we have very little understanding really of what the mechanisms of toxicity of nanoplastics are. There's inflammation that we believe is going to happen once they enter a tissue, right? So the body is just reacting to that foreign particle with an inflammatory response. There's oxidative stress production. There's quite many people demonstrating that. Now, oxidative stress is like a key node in a biological system. So everything, like, or basically almost everything, results in oxidative stress. And then there's this food dilution idea, just taking up space in the gut, and then you eat less, you get less energy intake. So we have kind of rough ideas of what the mechanism of action of nanoplastics would be, but to be honest, that's purely, or in many cases, really speculative and hypothetical. So something to really follow up.
I think to add on to that from Martin as well, and being not a biologist, but I think that when we looked at engineered nanomaterials, we tried to look on read across toxicity and looking at particle size, but it ended up really being the ROS production and oxidative potential, and also the dissolution of the material. So it was really material dependent as opposed to size dependent. Of course, keeping in mind that smaller particles may dissolve faster or to a higher extent, but it was really about the chemistry of the material. And so in that case, it might also be a little bit difficult to look at size-based metrics between engineered nanomaterials and plastics, because the chemistry is really the driving factor, at least in terms of engineered nanomaterials.
Thanks, Nanna.
So I think this. Go on, sorry, go on.
Sorry, just adding on to that, actually, I think like we also have to be a little bit careful about talking about the toxic mechanisms of nanoplastics, because I mean, nanoplastics is an extremely diverse group with, I guess, almost an infinite number of possible combinations of polymers and additives and sizes and aging states and so on. So I think also just as a little terminology issue that you know, talking about nanoplastics in itself, it might not be so meaningful.
It's
I think that's an important point that Martin has drilled into me.
Yeah, I was just like hitting on you, just not to hit you, but just to say, like, yeah, important point. Nanoplastic is not nanoplastic is not nanoplastic. And I think a lot of the confusion really comes from calling a diverse set of pollutants one name, you know, like it would be like Chelsea Rochman said that it would be like if we look at all pesticides as one thing, and nobody would think that is a logical thing to do. But for microplastics, we do it, for nanoplastics, we do it. So we cover this diverse suite of pollutants under one umbrella term. And that is, I think, part of the problem of simplification that we're having here. Stop talking now. Melissa, you wanted to say something.
That's good. I think that's a really, really important point. I guess the question with that is how do you then get that into policy and things like this, because obviously one term is much easier to understand, but I think it's a very good comparison to pesticides, as you say. So I think a lot of this talk has been about kind of really interesting thing about volume, but if we move on to the next part, which I think is kind of key as well, we don't actually know how much is in the environment in terms of nanoplastics due to kind of like the difficulties of measuring them. So I think that's also a very important part of the discussion, if we can move on to this as well, to talk about how much we think is in the environment.
Well, I don't think I'll be able to answer how much is in the environment, even though this is my next slide. So I have a little bit of a letdown there. But here I think that we were just trying to present this idea that, of course, particles are not alone in the microplastics, nanoplastics, are not alone in the environment. There's a lot of other particles that are also there. And so what do we think it is really special about nanoplastics or microplastics or plastic particles that are different than these other natural or other engineered materials? Because if we were to have a worm that is living in a soil and eating a lot of soil and sand particles, we're not going to freak out. But now if it is eating some plastic, then it may be cause for concern. So really, what are the physical differences that would happen there? And so, I mean, I think that this was more just showing, keeping an eye on the other different materials which might be in the environment, and again, trying to see what is specific about nanoplastics that could induce different or more harm than other colloids that are already there.
Maybe it's related to this. Sorry, no, go ahead. Were you going to talk about this, Denise?
No, this is Julien's slide.
Oh, all right. Sorry, Julien. I just wanted to mention that I would like, just a quick incidental, that I want to thank also all people that sent their questions, and I realize that due to the time we may not be able to answer them all, but there was an interesting point that Melissa and I noticed, is that one interesting question about well, we've just been talking about how much is there, and we don't really know. So perhaps it's even more difficult to answer. But how do we get rid of it? Do you know any effort specifically made towards removing plastic, microplastic, nanoplastic from the environment?
Well, now I'm glad it moved to Julien's slide. No, but I think
so yeah, I think
having
yeah, go ahead.
Oh, no, please go ahead.
No, please. I think I will go after you. Answer the question after you.
It's no deep insight, believe me, but I think that it's going to be difficult / impossible to remove nanoplastic or microplastic, or as we even see macroplastic from the environment in large quantities. And so it's really just better waste management strategies. So if we look at how microplastics and nanoplastics are getting into the environment, it is degradation of mismanaged plastic waste. And so, okay, it's maybe an easy and obvious answer, but I don't think that it's going to be realistically possible to retrieve nanoplastics once we're there, because we can't even do this with macroplastic.
Yes, and I think there is no, up to my knowledge, there is no system in every country that can remove nanoscale material from wastewater treatment plant. So I think we, as much as they are produced, they will be released in the environment. That can be very interesting with a slide. So it summarized what. So Nanna make a very good point just before. Thanks, Nanna, because you explained better than me in English, so I don't have to repeat it. But I think we talk about plastic toxicity. Plastic is toxic. Plastic can be, but so we talk about size, but the real point is, I think, is a composition of a with the size, with the shape, with all these parameters, because polystyrene, polyethylene, polypropylene, they have different behavior, they are made differently, they have different properties, they have different additive that can be used to formulate it. So the size can have very strong effect about how they will release additive according to the diffusion of the metals of a pollutant that is inside the matrix of a polymer. So I think when we say plastic is toxic, we have to be very careful, because plastic is composing, I think, 80% of our environment. So we have to be, I think, to be sure about what kind of mechanisms we want to look. If it is mechanisms based on size or based on the brominated flame retardant or other trace metal effect. So I'm not very sure about the toxic effect of nanoplastic based on just size. And I would like to maybe just, it's what in this figure we with Mark and Natalie, we model with the release of additive based on bleaching of additive based on size and based, which using typically mathematical diffusion, Fick's law, and other diffusion of materials in the aqueous or aqueous system. So my point was, so Martin also talked about, I think, the size effect about translocation versus microplastic and nanoplastic. This is what we put in this figure also. But when I started to work on nanoparticles, I think it was 2-3 years ago, people work on carbon nanotubes, for instance, and they try to give how the size of carbon nanotubes can affect the organisms in terms of translocation. And they realize that a nanotube that can have 20 micro and 1 nanometer thick can still translocated in the cell. So we have to be also, the size have to be, I don't know, at some point they have to be bracketed between like shape, or if at least 2 or 3 parameters. Is what we here in the figure we put in this bread point, the 5, which is the bioptake and translocation. We want to say that at some point when the plastic is degraded and release nanoplastic, and we start to see some translocation. That means that we have nanoplastic, and it's what you already say, Martin, and I enjoy your talk. And I also want to say that when we go down to size, it's very difficult to quantify nanoparticles. So to say how much we have in the environment is almost, I would say, not possible for now. Some people have methods to quantify in lab, of course, but to quantify the number of nanoplastic that you have in the environment is, I think, it's totally, for now, for me, it's in the cloud. We have no idea how we can do that. But we know that you can identify your presence. Now we have some different techniques, and we know that they are here. We know that if we detect these nanoscale materials in the environment, that means that we have enough material to be detected. And it is I think it is the first time where we can measure nanoscale anthropogenic nanoparticles in the environment at this in this different environment, that is open ocean, soil, and air, and other system. And I think it's very interesting to see, very scaring, of course, but interesting as a scientific, how will be in the next 10 days, maybe 10 days, but 10 years in terms of, amount of nanoplastic that we will find in the environment. So this is moving, I think, to the last part of, I think, about techniques to detect nanoplastic. But maybe yeah, you see, this is my point. Nanoplastic is difficult to detect, and people use proxies, and proxies are always used in different fields, not only in nanoparticles, but for every kind of experimental design. You have your standards and so I think we have various different strategy to make proxies. The one that you repeat what you have in the environment, the one that you want to absolutely trace in your design at trace level. And I think this is what Denise did with particles at the spherical one. And I think it's very, very interesting to use this particle to be able to know the mechanisms about translocation, detection, and behavior in the environment. So I'm not sure who is the next in terms of slide. I forgot.
We're finished. We're finished with slides.
Oh, that's great. So yeah,
congratulations.
Yeah, and in terms of proxies, I think in our lab we try to develop like proxies that are very more relevant or more close to what we observe in the environment in terms of heterogeneity of shape and size and composition. And because we want to understand the behavior of this particle in the environment where we detected this particle. So if for what we decide to use this proxy development, but I let the speak to Denise, and maybe she have other way to do it.
Yeah, well, I also really like your proxies. I hope that one day we can converge our methods, and then we'll have the ultimate, the particles to test. But yeah, we go a little bit in a different direction, where maybe some of the particles are not 100% relevant, as Julien said. Some of the particles are spherical, not all. We have also fibers and fragments and different size classes and different polymers. But there we use proxies, specifically metal, although others have used fluorescence or C13 labeling or so. So we can kind of circumvent the difficulty of analysis, because if we're anyways working in a lab system, we don't need to take the extra time to be able to then measure the particles afterwards, if we can more easily measure with a trace metals approach. And I think that this has allowed us to test a lot more processes more easily and quickly. But then again, it does come to the question about if you used materials with different surface functionality or chemistry or polymers, if it would change your result in the end. So I think that there's pros and cons on both sides about using materials which have a dopant in them versus trying to collect materials from the environment to use in your laboratory studies.
Well, Thanks very much. I have to say that we are approaching the end of the webinar, and there is so much more to say. I'm really sorry for all those very nice questions that have been sent and that I have here in front of me. Whether it would take too long to reply. I'm trying to save them though, and I'm going to send them to all the panelists just to add a little bit more work to the amount of stuff they have to do already. And who knows, maybe we can in some time do a second episode to discuss a little bit more, some more developments. But I'd like to thank everybody for attending. Thanks very much to Martin, Nanna, Julien, and Denise. Thank you, Mel, for your help in organizing this, and I hope everybody has enjoyed it as much as I did.
Thanks.
Thank you.
Thank you very much.
Bye.
Bye. Just before I go, I would like to remind all the attendees that this will be this is being recorded, so we will post it on our website very soon if you want to look at it again. Thanks again. Bye.